Hi students, இன்றைக்கி நம்ம பார்க்க போகிறது உங்களுடைய யூனிவர்சிட்டி ப்ராக்டிக்கல் பேட்டர்னில் இருக்கிற யூரின் அனாலிசிஸ். ரைட்டா? உங்களுக்கு யூரின் அனாலிசிஸ் சம் 30-40 மார்க்ஸ் வச்சுருவாங்க ஒரு பேட்டர்ன். இன்னொரு பேட்டர்ன் எஸ்டிமேஷன் ஆஃப் சம் காம்பவுண்ட்ஸ் லைக் குளுக்கோஸ், யூரியா, கிரியாட்டினின், புரோட்டீன் லைக் தட். இப்போ இந்த அப்நார்மல் கான்ஸ்டிடியூவன்ஸ் ஆஃப் யூரின்றது ரொம்ப ரொம்ப இம்பார்ட்டன்ட் ப்ராக்டிக்கல் பாயிண்ட். இந்த இதை நீங்கள் தெரிஞ்சுக்கிறதுக்கு முன்னாடி யூரினுடைய ஸ்பெசிஃபிக் ஃபியூச்சர்ஸ் ஆர் ஸ்பெஷல் கேரக்டர்ஸ் தெரிஞ்சுக்கணும். அதாவது ஸ்பெசிஃபிக் கிராவிட்டி எவ்வளோ, வால்யூம் எவ்வளோ, பிஹெச் எவ்வளோ, இதெல்லாம் தெரிஞ்சுக்கணும். நம்பர் 1 அபவுட் யூரின், நார்மல் யூரின். செகண்ட் திங் இஸ் யூ ஷுட் நோ அபவுட் தி நார்மல் கான்ஸ்டிடியூவன்ட்ஸ் ப்ரெசன்ட் இன் யூரின். யூரினில் நார்மலாக என்னென்ன காம்பவுண்ட்ஸ் இருக்குன்றதையும் தெரிஞ்சுக்கணும். நான் அதை இன்னொரு வீடியோவில் நார்மல் யூரின் அனாலிசிஸ் போட்டிருக்கேன். இப்போ நம்ம பார்க்க போகிறது அப்நார்மல் யூரின் அனலைசிஸ். இந்த அப்நார்மல் யூரின் அனலைசிஸில் 7-8 காம்பவுண்ட் கொடுத்துருப்பாங்க. பட் யூஸ்வலாக எல்லா யூனிவர்சிட்டிலையும் வைக்கிறது 5 மாலிக்யூல்ஸ் 5 காம்பவுண்ட்ஸ். சரி, அந்த 5 என்னென்னன்றது அதுக்கான டெஸ்ட்டு பார்க்க போகிறோம். இந்த டெஸ்ட்டு இதெல்லாம் அப்நார்மல் கான்ஸ்டிடியூவன்ஸுக்கு முன்னாடி நீங்கள் என்ன தெரிஞ்சுக்கணும்னா 5 டிசீசஸ் ரொம்ப ரொம்ப முக்கியம். அதாவது எந்த டிசீஸுக்கு நம்ம இந்த டெஸ்ட்டெல்லாம் பண்ண போகிறோன்றதை ஃபஸ்ட்டு உங்களுக்கு தெரியும். Number 1 diabetes, number 2 starvation, number 3 jaundice, number 4 hematuria, number 5 proteinuria. It's nothing but very simple. Diabetes, you know very well, hyperglycemia, glucose level அதிகமாக இருக்கும். Starvation, another extreme, no glucose in fasting state for a longer period. Jaundice, presence of bilirubin, excess bilirubin in urine means jaundice. Hematuria means blood or hemoglobin in urine. Blood irundaka hematuria, hemoglobin irundha hemoglobinuria. Last one is proteinuria. Protein urine irundhuchina proteinuria. Right? இந்த 5 டிசீசஸ்க்காக தான் நம்ம சில டெஸ்ட் பண்ண போகிறோம். அதில் என்ன ரிசல்ட் கிடைக்குது, அந்த காம்பவுண்டை வச்சுக்கிட்டு நம்ம என்ன டிசீஸ் அந்த பேஷண்ட்டுக்கு இருக்கலாம், ப்ராபபிள் டயக்னோசிஸ், மேபி, அந்த மாதிரி நம்ம சொல்ல போகிறோம். சரியா? உங்களை என்னென்ன கொஸ்டின் கேட்பாங்க, அந்த டெஸ்ட்டு பேர் என்ன, எப்படி நம்ம அது இப்போ ஆன்சர் சொல்லணுன்றதை இப்போ நான் 1 by 1 சொல்கிறேன். ஓகே, ஃபஸ்ட்டு டெஸ்ட் இஸ் Benedicts test. This is very 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 important test, Benedicts test. இப்போ இந்த கொஸ்டின் இந்த டெஸ்ட்டுக்கு வர எக்ஸாமினர்கிட்ட நீ ஆன்சர் சரியாக பண்ணலைன்னா டெஃபினட்டாக அது ஒரு பேட் இம்ப்ரெஷன். உனக்கு ஒன்றுமே தெரிலன்னு மீதி டெஸ்ட்டெலாம் உனக்கு நல்லா தெரிஞ்சிருந்தா கூட அது இட் வில் கிவ் பேட் இம்ப்ரெஷன். ஸோ யூ ஷுட் நோ தரோலி A to Z about Benedicts test. ஓகே, ஸோ Benedicts test எதுக்கு பண்ணுறோம்னா இட் இஸ் எ டெஸ்ட் ஃபார் ரெடியூசிங் சுகர். ரைட்? ஃபஸ்ட்டு கொஸ்டின் ஃப்ரம் யுவர் எக்ஸாமினர் இஸ் why you want to perform Benedicts test. It is a test for reducing sugar, to find out the reducing sugar. Right? Second one is Rothera's test. Why you want to perform? It is a test for ketone bodies. Third one is some places they are having for blood test hematuria like that benzidine. In some labs, some universities they are giving orthotoluidine test. I will discuss both tests in the next slide. Don't worry. So benzidine or orthotoluidine test, both are for presence of blood and hemoglobin. If blood is present in urine in hematuria, hemoglobin is present in urine means hemoglobinuria. Next one is Hayes test, H-A-Y-S, Hayes test. It is a test for bile salts. Next is heat coagulation. It is a test for proteins. So புரோட்டீன் இருந்தால் என்ன டிசீஸ் சொல்ல போகிறீங்க, பைல் சால்ட் இருந்தால் என்ன டிசீஸ் சொல்ல போகிறீங்க, பிளட் இருந்தால் என்ன டிசீஸ் சொல்ல போகிறீங்க, கீட்டோன் பாடிஸ் இருந்தால் என்ன டிசீஸ், அண்ட் ரெடியூசிங் சுகர் இருந்தால் என்னென்ன டிசீஸ்ன்றதை நம்ம சொல்ல போகிறோம். ரிசல்ட் எழுதும்போது நீங்கள் ப்ராக்டிக்கல் ப்ரொசீஜர் முடித்து கடைசியாக ரிசல்ட் எழுதும்போது இதெல்லாம் சொல்லணும். இப்போ அது எப்படி எழுதணுன்றதுக்கு ஒரு ஹிண்ட்டு கொடுக்குறேன். சப்போஸ் உங்கள் டெஸ்ட்டோடைய ரிசல்ட் ரெடியூசிங் சுகரும் பாசிட்டிவாக இருக்குது, ப்ரோட்டீனும் பாசிட்டிவாக இருக்குன்னா there are so many conditions, but for you, that is undergraduate students, only one diagnosis is diabetic nephropathy. You got it? Your result will be, suppose reducing sugar positive and protein test, that is heat coagulation test positive, means your answer should be diabetic nephropathy. Like that, reducing sugar present and also ketone body present means the patient is having diabetic ketoacidosis. Right? Usually they won't give single urinary reducing sugar for diabetes. That is why I am giving either diabetes or diabetic ketoacidosis. Mostly they will be giving both reducing sugar and ketone bodies. The answer is definitely diabetic ketoacidosis. Don't write your result single reducing sugar only present. Along with reducing sugar, definitely ketone bodies will be there or protein will be there. If the protein is there, means the condition is diabetic nephropathy. If the decay, that is ketone body is present, means diabetic ketoacidosis. Right? Third one is bile salts positive and bile pigments positive means jaundice. And blood is present along with the protein. The answer is AGN, that is acute glomerulonephritis, one of the kidney problem, that too in young age group. Patient or babies are having blood in urine hematuria, protein in urine proteinuria, means the diagnosis will be acute glomerulonephritis. Right? Hi. For this Benedicts test, what are all the questions? Repeatedly they will be asking in your university exam practical time. Whoever may be the examiner, MGR University, Karnataka University, Andhra University, Pondicherry University, everywhere the questions are same. You should know the answer for all the questions. Definitely only 2 or 3 questions only they will be asking and give marks to you. Okay. First question is, it is a test for what? The name of the test is Benedicts. It is a test for reducing sugar, to find out the reducing sugar. Right? Second one is, what is reducing sugar? You have to give 1 or 2 lines of definition. Carbohydrates having group, right, or either aldehyde group or keto group, which exhibits reducing property, right? That is known as reducing sugars. They are having reducing property. Okay, aldehyde group or keto group. Third one is, you have to give examples. In the examples part, you keep it in mind, you have both monosaccharide and disaccharide coming under examples of reducing sugar. Right? Monosaccharide means glucose, fructose, mannose, galactose, like that. Disaccharide means lactose and maltose. Reducing sugar. Okay. Second one is, what is the principle? How this Benedicts reaction occurs? It is very simple. Copper sulfate, one of the important in that Benedicts reagent, one of the important compound is copper sulfate. This copper sulfate releases cupric ions. Okay, first one is cupric ion, Cu2+. These cupric ions combined with enediol. Enediol means in an alkaline medium, you are having some carbohydrates for a longer period, they will go for tautomerization and releases some compound, that is enediol. This cupric ion combined with this enediol releases cuprous ions and some sugar acid. This cuprous ion, that is Cu+, 1+, releases 2 cuprous hydroxide molecules, which releases cuprous oxide and water molecule. Right? Very simple, only one reaction, very simple. But some examiners, they will ask what is the principle means. First word is enediol formation. Second is copper sulfate converted into cupric ions, cuprous ions, and finally cuprous oxide. Cuprous oxide only will give the color for the Benedicts reaction. Okay. So this is regarding the principle. What is the procedure? Very easy procedure. You have to take the test tube. First, in Benedicts test, first you have to take 5 ml of Benedicts reagent. Some colleges, they are asking about to take 3 ml. Some places they ask to take 5 ml. That is reagent only. Another one is, you are having urine solution. You are going to identify which compound is present in that urine solution. So first you have to take 5 ml of Benedicts reagent. Then you have to add 8 drops of urine sample. 8 drops of urine sample. Okay. Then you have to boil it for 2 minutes. This heating time is very important, 2 minutes. Why you don't want to go for 1 minute? Why you are not heating 3 minutes? Means suppose the examiner asks means, if suppose your solution is having disaccharide, it will be dissociated into 2 monosaccharides and results will be false positive. If you are having monosaccharide means no issues for 1 minute, 2 minutes, 3 minutes. Whereas if your solution is having disaccharide, after 2 minutes what will happen is, this disaccharide is splitted into 2 monosaccharides and will give positive result. That will misguide you. Your solution is monosaccharide. Actually your solution is having disaccharide. This is very important. Heating time. Okay. Then you have to wait for the color change. Finally you are getting 4 colors. Your original test Benedicts reagent color is blue. After heating this Benedicts reagent with the glucose urine solution, you will get 4 colors like rainbow color, GYOR. In the WIPGR you have to take the last 4, green, yellow, orange, red. In this 4 you will get definitely 1 color, depending upon the concentration of glucose in urine. How will you identify the concentration? We have one small formula, that is, if it is green color means your glucose concentration starts from 0.5 to 1 gram, 1%, right? Second, yellow means 1 to 1.5%. Orange means 1.5 to 2%. If it is red means more than 2%. Sometimes some books says red, some books says brick red. Both indicates more than 2% of glucose present in urine. In another method is green means 1 plus, yellow means 2 plus, orange means 3 plus, red means 4 plus. In your requisition form they will mark like this, 1 plus, 2 plus, 3 plus, 4 plus, like that. After receiving the report, the clinician will interpret depending upon the glucose concentration how the patient's result. Okay, this is regarding the procedure. Next one is color and glucose concentration. I already told you. GYOR. What are all the colors? Green, yellow, orange, red. Next one is, what is the composition of Benedicts reagent? Most of the examiners ask about the composition. If you are telling the composition, definitely they will move on to next question. Sodium carbonate, sodium citrate and copper sulfate. Right? Don't forget. What is the role of these 3? Copper sulfate releases cupric ions. Sodium carbonate gives alkaline medium. For any reaction you have to go for either alkaline medium or acid medium. This sodium carbonate produces alkaline medium for Benedict's test. In this alkaline medium only they will go for enediol formation. Third one is sodium citrate. It only prevents the precipitation because the reagent compounds only will get or will produce some precipitation. This sodium citrate prevent the precipitation and keep the cupric ions available for the Benedict's test. So what is the composition? Sodium carbonate, sodium citrate and copper sulfate. You have to tell all the 3 reactions for each, functions for each. Right. Next one is enediol formation. I already told you, this is the benzene ring in 1 double bond, 2 hydroxyl group attached. This is the structure of enediol formation. Next is, what is enediol? Already I told. Example for reducing sugar, monosaccharide and disaccharide. Here NRS means non-reducing sugar. We are having only 2 non-reducing sugars. One is sucrose, another one is trehalose. TREHALOSE, trehalose. These 2 are coming under non-reducing sugar. Right? This question is very important. Another one is, sometimes what happens is some non-carbohydrate substances. See, monosaccharide, disaccharide, sucrose, everything coming under carbohydrate substances. In some places, some reactions, some non-carbohydrate substances also will give Benedict's positive. So we have to keep it in mind to rule out or interpret the investigation report. These are number 1 ascorbic acid, number 2 homogenesic acid, next is salicylates and glucuronic acid. So there are so many compounds. I am giving just 3 acids: ascorbic acid, glucuronic acid, homogentic acid, and salicylates. Right? These are all the compounds or non-carbohydrate substances which will give positive Benedict's test. Okay? Then why it is semi-quantitative? Why it is not a specific test? Why it is semi-quantitative means it will give the result of what is the amount of sugar present in your urine approximately. That is why this is a semi-quantitative, not a specific one. This result will not reflect perfect amount of glucose present in urine. That is why semi-quantitative. That is why not a specific test. Okay. Next, how will you write the result? The given solution contains reducing sugar, carbohydrate, monosaccharide, either glucose or fructose. You got it? The given solution is carbohydrate. The given solution is disaccharide or monosaccharide. The given solution is reducing sugar or non-reducing sugar. The given solution is either sucrose or glucose or fructose. Like that, you have to specifically mention the result. Okay. If it is glucose means very simple: carbohydrate, reducing sugar, monosaccharide, sugar, glucose. Okay. So this is the way you have to write the explanation. Finally, suppose if your result is 1 plus 2 plus 3 plus 4 plus, you have written like my given solution contains monosaccharide or disaccharide, like that. The final question will be, in which conditions you will get glucose in urine. There are so many diseases. I am giving only 3. First one is diabetes. For all diabetic patients, either 1 plus, 2 plus, 3 plus, 4 plus of sugar will be present in urine. Right? Diabetes mellitus. Second one is actually renal glycosuria. Also, it is here in the answer for conditions for glucose in urine is renal glycosuria and diabetes mellitus. Third is gland involvement. Some diseases, endocrine diseases, adrenal diseases, pituitary diseases, thyroid disease, that is hyperpituitarism, hyperthyroidism, hyperadrenalism. The patients will have high amount of glucose, that is hyperglycemia. Definitely they will get glucose in their urine. The last one is steroid. Steroid is always stimulating the glucose metabolism and they will go for hyperglycemia. So this is regarding Benedict's test. Like that, we are going to discuss all the 5 tests one by one. Okay.